Hello, I am Kevin Kim, and I recently joined OliX. I earned my bachelor's degree in neuroscience and a PhD in pharmacology at UCLA, and have since built my drug discovery career at various research institutions and pharmaceutical companies. Most recently, I have been spearheading multiple RNA therapeutics programs. At Wave, I directly led numerous CNS therapeutics programs. At Sarepta, I served as senior director overseeing the discovery biology group, and at a startup called MeRecuel, I oversaw the entire research team as vice president. Leveraging my expertise in CNS, cardiovascular, muscular, and renal diseases, alongside my extensive experience in developing diverse modalities of RNA therapeutics, I look forward to successfully driving OliX's new RNA drug discovery programs forward. Today, I would like to share an overview of our CNS and extrahepatic platforms. Previously, OliX had established its own proprietary siRNA technology platform for the CNS, focused on delivering siRNA therapeutics via direct intrathecal administration. Our first-generation OaSIS CNS platform is able to achieve uptake in multiple cell types and distribute to different regions of the CNS. When targeting a gene expressed widely in the CNS, our platform demonstrates robust potency in knocking down the target gene in various CNS regions in a dose-dependent manner. In addition, our proprietary siRNA platform demonstrates a significant duration of effect, lasting more than 3 months with a single intrathecal injection. While the platform we have built so far is highly effective, we, as well as others in the field, recognize the lack of uniform distribution and efficacy of the injected siRNA with this administration method, particularly for the deeper brain regions. In addition, an intrathecal injection via lumbar puncture is a highly invasive procedure that limits patient convenience and may lead to reduced compliance. As an alternative to this invasive procedure, the field has been working to develop methods to deliver drug moieties to the brain via systemic administration, such as intravenous or subcutaneous injections. Our brain and spinal cord are enveloped by a vast network of blood vasculature that supplies oxygen and nutrients to the CNS. However, to prevent toxins from entering the CNS, our body has a protective system called blood-brain barrier, or BBB, that acts as a security gate, allowing vital nutrients like oxygen and glucose to pass through to the brain while blocking harmful toxins and bacteria. Due to the BBB, over 98% of large molecules are blocked from entering the brain, and this blockade is currently recognized as one of the major challenges in developing therapeutics for CNS diseases. To overcome these delivery limitations, scientists in both academia and industry have been researching ways to utilize the internal transport system, specifically receptor-mediated transcytosis. By leveraging receptors such as transferrin receptor 1 or TFR1, we are now able to deliver large molecules, including antibodies and siRNA, from the bloodstream through the BBB and into brain cells. This method has been proven to achieve more uniform drug delivery throughout the CNS, including deep brain regions. Utilizing this endogenous transport system enables systemic administration, namely subcutaneous or intravenous injection, thereby enhancing patient convenience. Today, multiple therapeutics targeting TFR1 have been approved or are undergoing active clinical trials. Among them, Arrowhead Pharmaceuticals, an RNAi company, is conducting a Phase 1 and 2A clinical trial for an Alzheimer's disease treatment that delivers siRNA via TFR1-mediated transcytosis. As part of the OliX 2.0 strategy, we have initiated our efforts to develop siRNA drugs that can be delivered via less invasive systemic administrations by partnering with leading BBB shuttle technology companies such as Vectoris. In this collaboration, we are utilizing Vectoris's well-validated VHH nanobody technology, targeting transferrin receptor, to deliver the siRNA payload from the blood to the brain cells. By covalently joining BBB penetrant antibody to OliX's CNS-validated siRNA, we are able to generate a new complex modality known as antibody siRNA conjugate, or ARC. Today, we would like to give you a brief update on our progress on the development of our ARC platform. To evaluate the competitiveness of our second-generation OliX CNS platform, we analyzed the brains of mice 2 weeks after administering 3 doses of 1.5 milligrams per kilogram via either intravenous or subcutaneous injection. The results confirmed that our ARC significantly knocked down the target gene across various brain regions, including deep brain structures such as the striatum and hippocampus. The study demonstrated a knockdown efficacy of over 70 to 80%, which is comparable to data from a competitor's platform, indicating that our ARC performs at an equal or superior level to the competition. A key point to emphasize is that the subcutaneous administration yielded a knockdown profile similar to that of intravenous administration. This opens up the possibility of designing CNS therapeutics with the most ideal route of administration for patients in the future. While our CNS disease programs are currently in the early discovery stage, we would like to briefly introduce our approach to drug discovery. Through our first-generation OliX CNS platform, we successfully identified a lead siRNA that downregulates target genes known to induce CNS diseases in human cells. Then, utilizing intracerebroventricular administration as a local delivery method, we confirmed the in vivo efficacy in mice. Furthermore, using a mouse CNS toxic aggregate model, we confirmed the potential therapeutic efficacy of these lead siRNAs in reducing toxic accumulations. And recently, we evaluated a second-generation ARC molecule, which combines a lead siRNA discovered from our first-generation OliX CNS platform with Vectoris's VHH antibody. Following intravenous or subcutaneous administration in mice, we confirmed a significant level of gene knockdown efficacy in disease-relevant regions. While there are limitations to a direct head-to-head comparison, the mRNA knockdown levels achieved through systemic administration appear comparable to those of local administration. Based on these promising results, we are now well-positioned to leverage less invasive, yet equally effective systemic ARCs to develop potential therapeutics for patients with CNS diseases. OliX has already made significant strides in its new innovative strategy to develop meaningful therapies for CNS diseases using ARC, our second-generation platform. In addition to collaborating with antibody developers equipped with proven BBB penetrant systems, we are enhancing our platform by leveraging AI technology for next-generation antibody development. To ensure successful drug development, we will establish optimized ARC modalities and scalable manufacturing processes, while evaluating their duration of effect and tolerability in mouse models. Furthermore, as our CNS programs advance, we plan to validate our platform using NHP as CNS programs progress. Moreover, OliX possesses the robust capabilities required to improve our current ARC technology by utilizing proprietary in-house platforms, such as previously mentioned dual targeting platform. Leveraging this cutting-edge technology, we are dedicated to developing truly effective and meaningful therapies for CNS patients. To achieve this, our pipeline strategy focuses on targeting not only the validated genetic targets, but also the targets in core pathologic pathways implicated in each condition to advance our siRNA therapeutics. Additionally, as part of our extrahepatic tissue targeting strategy, we are currently developing platforms for the kidney and muscle. The kidney is currently one of the most prominent areas of focus in the field, drawing significant early-stage investments and active capital from various pharma and biotech companies. Historically, the kidney has been recognized as an organ where oligonucleotides, particularly antisense oligonucleotides, accumulate, making it an excellent candidate for nucleic acid therapeutics. However, it is composed of a complex architecture with 40 to 50 distinct cell types, each performing critical functions. Consequently, a targeted delivery vehicle capable of selectively reaching specific cell types is essential. To achieve this, we are actively screening a diverse pipeline of peptides and lipids, For muscle targeting, technologies leveraging various vehicles, such as lipids, peptides, small molecules, and antibodies to efficiently deliver oligonucleotides into muscle cells have already been broadly validated. Active clinical trials are currently underway for muscular dystrophies like DMD, DM1, and FSHD. At this stage, rather than selecting a single vehicle, our strategy will focus on specific disease indications and matching each with the most suitable delivery vehicle. For instance, because the market for rare genetic diseases with well-defined targets is already intensely competitive, the most efficacious, best-in-class vehicle must be deployed. Conversely, multisystemic disorders like DM1 require a vehicle capable of multi-organ delivery. Furthermore, to prevent muscle loss in the rapidly growing market for weight loss programs, utilizing a cost-effective vehicle is preferable to ensure high market accessibility. To this end, OliX is currently evaluating and profiling multiple delivery systems to select the most optimal vehicle for each specific disease. Leveraging our years of experience, proven success, and global expertise, OliX Pharmaceuticals will continue to establish various extrahepatic platforms and develop the most appropriate siRNA therapeutics for patients in urgent need. Thank you for your attention.